In double fertilization, after the pollen land on the stigma, a pollen grain germinates and produce pollen tube. Okay, so this pollen tube will extend down to the cells of the stele and go to the ovary. The pollen tube discharge two sperm, okay, which are previously produced by the generative cell. The sperm are discharged into the embryo sac. Then one sperm will fertilize the egg, form a zygote, and the other sperm will combine with the two polar nuclei and form triploid nucleus of endosperm. So these are the image of the two sperm nuclei which are going to the embryo sac in female gametophyte. So this is the process. Number 1, if a pollen grain germinates, a pollen tube grows down the stele to the ovary. The pollen tube discharge two sperm. One will fertilize the egg, the other will combine with the two nuclei, okay, polar nuclei. A seed consists of an embryo, a nutrient supply, and a seed coat. In double fertilization, after the process, each ovule develops into a seed. Meanwhile, the ovary later will develop into a fruit. Okay, for embryo development, the first mitosis of the zygote is in the form of transverse. Okay, so the process will split the fertilized egg into a basal and terminal cell. So this is a zygote. Okay, so they do the transverse mitosis division, form terminal cell and the basal cell. Endosperm development usually precedes the embryo development. Okay, precede means come before. So the endosperm development first happen, later followed by the embryo development. In most monocots and some dicots, eudicots also known as dicots, the endosperm will store the nutrient that can be used by the seedling after germination. Okay. Meanwhile, for other dicots, the food reserved by the endosperm will completely export to the cotyledons. Now we go into the structure of the mature seed. Okay, the embryo and its food supplies are enclosed by a hard protective seed coat. So the character of the seed coat is hard because it is functioned to protect the seed. In a common garden bean, okay, a eudicot or dicot, the embryo have two cotyledons. Okay, and the embryo also consists of the hypocotyl and the radicle. Okay, the radicle here. Okay, and the thick cotyledon. So this is the structure of the mature seed in garden bean. Okay, meanwhile, this is the seed of other dicots such as castor bean. In castor bean, the cotyledon is quite narrow and membranous. Okay, so quite narrow. Okay, and then it absorbs food from the endosperm. So this is the endosperm, the food storage, okay, from the endosperm when the seed germinates. Okay, the embryo of a monocot. So this is monocot, meaning they have one cotyledon. A coleoptile and coleorhiza. Okay, they have single cotyledon with coleoptile and coleorhiza. Okay, so this is the coleoptile and this is the coleorhiza. Okay, so this can be found in maize. Okay, like all monocot, maize has only one cotyledon. Maize and other grasses have a large cotyledon called scutellum. Okay, so the cotyledon is known as scutellum. The rudimentary shoot is sheet in a structure called the coleoptile, and the coleorhiza cover the young root. Okay, so the shoot is covered. Okay, the shoot is covered by the coleoptile, and the coleorhiza is cover the root. Okay, from the ovary later form a fruit. So the fruit developed from the ovary, the function is to protect the enclosed seed and fruit also aids. Aids mean help in the dispersal of seed by wind or animals. So these are the examples of fruits. Okay. Fruits are classified into several types, depend on their developmental origin. Okay, for example, simple fruit. A simple fruit developed from a single couple or several fused couple of one flower. For example, the pea, lemon, and peanut. And we have aggregate fruit. Okay, aggregate fruit meaning more than one fruit. Okay, in a group, developed from many separate couples of one flower. For example, raspberry, the blackberry, and the strawberry. And the last is multiple fruit. Okay, this multiple fruit is developed from many couples of many flower. Okay, for example, pineapple and fig. So these are the differences in terms of the types of fruit. We have simple, we have aggregate, and multiple fruit. Okay, now we go into seed germination. Okay, as a seed mature, it dehydrates and enter a phase that referred as dormancy. Okay, dormancy is like resting stage of a seed. So this is happen when the condition is extremely low metabolic rate and the suspension of growth and development. So there is a suspension. Suspension meaning there is no growth and no development happen for the seed. Okay, so we call the seed is in dormant stage. So seed dormancy is an adaptation for tough times. Okay, when the environmental condition required to break dormancy. Okay, normally this seed dormancy happen when there is non-favorable environmental condition. Okay, so with the condition of seed in dormant stage, it will increase the chance that germination will occur at the correct time and correct place. Okay, which will give advantage to the seedling. Okay, when there is correct time and the place is good or favorable for the seed to germinate, the seed will germinate at that time. The breaking of seed dormancy often require environmental cues such as temperature or lighting cues. Okay, so in lab, for example, we can break this seed dormancy artificially, meaning we can expose them to the change in the temperature, or maybe we can give them lighting in order to break the seed dormancy period. The length of time of dormant seed remain viable and capable of germinating varies, either from few days to decades or even longer. So this depends on the seed itself. So the seed can be dormant either in just few days or maybe some decades or even longer than that time. So depending on the plant species and the environmental condition. Okay, from seed to seedling, germination of seed depends on the physical process called imbibition. Imbibition is the uptake of water due to low water potential of the dry seed. So imbibing water causes the seed to expand and rupture its coats and trigger the metabolic changes in the embryo that enable it to resume their growth. Okay, so once the seed coat is ruptured, then they can start to grow. So this is the process of seedling. Okay, from a seed and then they just start to germinate. Okay, seed germination or epigyle. The radicle is the first organ to emerge from the germinating seed. Okay, so remember the radicle part. In many eudicots, a hook forms in the hypocotyl and growth pushes the hook above the ground. So please identify which is the radicle. So this is the radicle. Later will form the root and the hypocotyl. Okay, in the form of hook here. Okay, hypocotyl. Okay, so this process is stimulated by light. The hypocotyl will strengthen, raising the cotyledon and the epicotyl. Okay, so you can read the process of epigyle germination. Okay, now we go into hypogea. Okay, monocot use different method for breaking ground when they germinate. Okay, so this is the germination for monocot. We call them as hypogea. The coleoptile, okay, so they have coleoptile and coleorhiza. Okay, the coleoptile, the sheets that enclosing and protecting the embryonic shoot will push upward through the soil and into the air. So this is the coleoptile, will be pushed over the ground. Okay, so this is happen in maize. Okay, now we go into plant growth. There are two types of plant growth, the primary growth and secondary growth. In primary growth, it involved with the lengthens of root and shoots. For secondary growth, it involved with the add girth to stem and roots in woody plants. Add girths meaning the plant is increased in their thickness or the diameter. Okay, so this is the structure of a plant. So we see the first is primary growth in stem. Okay, so meaning the stem is grow in length. Okay, they become longer. Alright, but for secondary growth, they become increase in diameter or thickness. Okay, so that is the difference between primary and secondary growth. So these are the overview of primary and secondary growth. Okay, if you can see the primary growth, okay, cell normally including the epidermis, cortex, primary phloem, primary xylem, and the pith. Okay, so normally the plant, like herbaceous plant, they do only primary growth. But for woody plant, normally they can have both the primary and later they will go for secondary growth, where they increase in their diameter or their thickness. So if you can see here, the difference is they have the cambium that act the secondary dermal tissue. Okay, and then this cambium later will form the secondary phloem and the secondary xylem. Okay, so that's why they can increase in their diameter. From the vascular cambium here, they will later form the primary xylem and the secondary xylem and the secondary phloem. Okay, now we go into the primary growth, lengthen roots and shoot. So this is the normal plant development we call as primary growth, where the plant is become longer or lengthen in their root and also their shoots. Primary growth produce the primary plant body, the parts of the root and the shoot system, and produced by the apical meristem. Apical meristem can be found at the apex. Apex meaning at the end of the shoot and at the end of the root. Okay, it is like stem cell, okay, that can produce plant cell. Okay, so the primary growth of roots, okay, the root tip. So this is the structure of root. Okay, the root tip. Okay, the root tip normally covered by a root cap. Okay, so this root cap which will protect the delicate. Delicate mean the structure of the root apex is very soft. Okay, so the root cap will protect the delicate apical meristem as the root pushes through the harsh soil during the primary growth. So that is the function of the root cap. Okay, the primary growth of roots, they produce the epidermis, the ground tissue, and the vascular tissue. So vascular tissue is very common. You already know the xylem and phloem. So this xylem and phloem is actually belongs to vascular tissue. For ground tissue, normally you used to know on parenchyma cell, collenchyma cell, okay, and sclerenchyma cell. So these three cells are belongs to ground tissue. And the epidermis, we have two types. Okay, the epidermis, we have the periderm and epidermal. Okay, so this is the organization of primary tissue in young roots. Okay, the epidermis, which is the outer layer, and then the cortex, the vascular, okay, in the middle, and also the core of the parenchyma cell. Okay, parenchyma cell is belong to the ground tissue. Xylem and phloem belongs to the vascular tissue. So this the image under microscope. Okay, you may see a more clear image. Okay, so if you can see, the cell here is stained by colors. So that staining is actually used to label each of the organ. Okay, now we go into lateral roots. Okay, lateral roots, this is the main root, this is the lateral roots, okay, which form at the side of the main root. Arise from within the pericycle. So it arises from the pericycle, the outermost layer in the vascular cylinder. So this is the vascular cylinder, number 1. Okay, and the number 2 image, it start to grow lateral root, and then the lateral root is protrude out. This is the primary growth of shoots. Okay, so this is the apical meristem in shoots. Okay, later this will form the leaf primordia, will form the leaf. Okay, so this is the early structure of the leaf before it form into complete leaf. Okay, a shoot apical meristem is a dome-shaped mass of dividing cell at the tip of the terminal bud. So this will give rise to a repetition of internodes and leaf-bearing nodes. Okay, if you still remember the structure of plant, okay, between one leaf to another leaf, okay, this is called the internodes. Okay, this is one leaf. Okay, between one leaf to one leaf is called the internodes. Okay, the structure that hold the leaf we call as node. Okay, so one node to another node. So between one node to another node is internodes. Okay. Alright, tissue organization of stem. In most eudicots and gymnosperm, okay, the vascular tissue consists of vascular bundle that arrange in a ring. Okay, so this is the vascular bundle. Okay, they form in the form of ring. So the pattern of the vascular bundle is ring shape. Okay, now we go into monocots. Stem. The vascular bundles are scattered. Okay, scattered meaning they do not have a rigid form of arrangement. Okay, so the vascular bundle in monocots are scattered. Meanwhile, the vascular bundle in eudicots is arranged in a ring. So that is another characters that can differ between monocots and dicots. Okay, this is about the tissue organization of leaf. Okay, the epidermal barrier in leaf is interrupted by the stomata. Okay, so we have stomata underneath the leaf. So the function of stomata to allow the carbon dioxide exchange between the surrounding air and the photosynthetic cells within a leaf. The ground tissue in a leaf is sandwiched. Sandwich means is layered between the upper and lower. So this is the upper epidermis. This is the lower epidermis. So in between of them are ground tissue. Okay, ground tissue mainly function for support system, and some of them also provide nutrition or storage for the plant. Okay, the ground tissue, if you still remember, there are 3 types of cells in ground tissue: the parenchyma cells, the collenchyma cells, and the sclerenchyma cells. And then inside the leaf also vascular tissue, which is continuous with the vascular tissue of the stem. Okay, so it is continuous with the stem and then go to the leaf. Same system. So this is the structure of the leaf tissue. Okay, so this is the upper epidermis, this is the lower epidermis. Okay, in between them are the ground tissue, which consists of the sclerenchyma, okay, parenchyma, and also the collenchyma cells. Right, and then we also have palisade mesophyll, spongy mesophyll, veins, the gut cells. Okay, which can be found control the stomata opening, and xylem and phloem is the vascular tissue. Okay, so all of these are ground tissue and vascular tissue which can be found in between of the epidermis. Okay, right, now we go into secondary growth, okay, which add girths to stem and roots in woody plants. So for secondary growth, it occurs in stem and roots of woody plant but rarely in leaf. The secondary plant body consists of the tissue produced by vascular cambium and the cork cambium. So you must remember, this secondary growth came from the vascular cambium and cork cambium. But for the primary growth, it happened in the apical meristem of the shoot and root. So these are important. Primary and secondary growth of a stem. So this one you may revise by yourself. Okay, look at the image and identify each part of the plant. Okay, this is again another diagram, okay, that shows the primary and secondary growth in stem. Okay, so this is the vascular cambium, which related to the secondary formation of the secondary phloem and secondary xylem. Alright, so it states here the vascular cambium is a cylinder of meristematic cells. Meristematic cell is meristem like undifferentiated cells, so it can differentiate into xylem or phloem later. So the cell is one cell thick, means it is very thin, developed from the parenchyma cells. Okay, parenchyma cell is one of the ground tissue. Okay, if you can see here, this is the secondary phloem, okay, which arise from the cork cambium. Alright, so later this secondary growth is actually will form the bark of the plant. The vascular cambium and the secondary vascular tissue. As a tree or woody shrub ages, the older layers of secondary xylem, the hardwood, no longer transport water and minerals, so they become dead cells. The outer layers, known as sapwood, the sapwood will still transport material through the xylem. So this is the sapwood, okay, which still living. And we have hardwood. Okay, hardwood is no longer function as a vascular system. Okay, secondary plant body. So the bark consists of the secondary phloem and the layers of periderm. So this is the vascular cambium. It produces sapwood. Sapwood is still function to carry the water and minerals, but hardwood is not function at all. And we have vasculary and the growth ring. Okay, this is another image of the 3-year-old of Tilia stem. Okay, now is the cork cambia and the production of periderm. Periderm is the epidermis of the woody plant. Okay, for woody plant, the epidermis is called periderm. So the cork cambium and periderm and the bark. So this, if you see the cork cambium, periderm and bark, you must know this belongs to secondary growth. Cork cambium gives rise to the secondary plant body protective covering or the periderm. Okay, the periderm consists of cork cambium plus the layer of cork cells it produces. Meanwhile, the bark consists of all the tissues external to the vascular cambium, including the secondary phloem and the periderm. Okay, so these are the references that is used for this chapter. If you have problems or difficulties in understanding this plant chapter, please refer one of these references. Okay, that's the end of this chapter. I hope you guys can revise from the lecture notes that is given in the Microsoft Teams. Thank you for listening.